In this next video, what we're going to talk about is when the patterns of inheritance don't simply follow what we discussed for plain old single gene inheritance. Because what we discussed in the previous video was with respect to single gene inheritance on the autosomes. And autosomes are any of the chromosomes that are not the sex chromosome. So for humans, which is what I'm going to primarily focus on in this video, autosomes includes chromosomes numbers 1 through 22, right? So that's 1 through 22 as you see on the karyotype in this slide. And if you don't know, a karyotype is just a chromosomal chart where the chromosomes, which are usually taken from the metaphase stage in cell division, they take them from metaphase and they lay the chromosomes in order from largest to smallest, but they usually put the sex chromosome pair at the end, chromosome pair number 23. So you can see how they're laid out in pretty much by size. And when it comes to genes that are on the sex chromosomes, right? So for females, the sex chromosomes are 2 X's. They are a homologous pair. For males, it's the X and the Y, which are a non-homologous pair. But we'll talk more about that later. But if we're talking about genes that are found on the sex chromosomes, the patterns of inheritance do not follow the same rules as they did when we were discussing autosomes. So this here is a pretty cool picture of the human sex chromosomes. It's displaying both the X chromosome right over here and the Y chromosome. You can see that they have been duplicated, so they're bearing their sister chromatids, which is kind of cool. But the other cool thing I wanted to point out about this photo is that you can see the major size difference between the sex chromosome pair in humans. Specifically, the X chromosome has about 1,500 genes on it, whereas the Y chromosome only has about 231 genes on it. So you can see that there's a huge difference when it comes to the amount, or when it comes to DNA content on sex chromosomes in humans. And those sex chromosomes are extremely important for sex determination. So again, this illustrates human development. So in early embryos, this is about week 5, we all develop unspecialized gonads. That's over here, right? Those 2 circular structures, which could either go on to form testes or ovaries. But right now, they're unspecialized. You have that, and you also have 2 sets of ducts. And so we see here the Wolffian duct and the Mullerian duct. So you got both of them. And those hang around until about week 6. Then after week 6, the decision of what to form, what those gonads are going to become, comes from information on the Y chromosome. There is a special gene called the SRY gene that encodes for a transcription factor. So the SRY gene makes a transcription factor. Transcription factor that will turn on male-specific genes. So if you have the Y chromosome, you should have the SRY gene. And so if you have the SRY gene, you're going to follow this path right here. Okay? What will happen is the gonads will become testes, and the Mullerian duct will be destroyed. Because the Mullerian duct, if it's allowed to stay around, it'll go on to form the fallopian tubes. But since we're going to be a boy, if we have the SRY gene, which is on the Y chromosome, then we destroy that duct. The other duct stays, which is the Wolffian duct, and that goes on to form the vas deferens, right? So now you got testes and you've got the vas deferens. You're going to turn into a male. However, if you do not have the SRY gene, then you do not have the transcription factors to turn on the male-specific genes. And so instead, the Wolffian duct gets destroyed, and you have the Mullerian ducts staying around. So those persist, and they go on to form the uterine or fallopian tubes, and the gonads go on to become ovaries, and you become female. So for us, The Y chromosome is hugely important for determining whether or not you're going to turn into a male or a female. The presence means you're going to develop into a male. The absence means a female. And if we look at this cartoon of the human sex chromosomes, there's some interesting things about them. So as we talked about in meiosis, all of your homologous pairs have to line up with each other during meiosis to ensure equal segregation, right? To ensure that half of the homologous chromosomes, each member of a pair is going to segregate to one side, and then the other member of the pair is going to go to the other side. So that way you produce good sperm and you produce good eggs. But if you're a male and you have an X and a Y for your sex chromosomes, they are not a homologous pair. Girls have XX, which is a homologous pair. Guys don't. So what corrects for that, so they can still find each other in prophase 1, right, and line up together in metaphase 1, what helps this out are two regions that are found on both the X and Y. They're called pseudo-autosomal regions. There's one up here, one down here. And they are both present. They're present on both the X and the Y chromosome. And if you look at the name, pseudo-autosomal, this implies that these regions of the chromosomes act like autosomes, like non-sex chromosomes. And what they mean by that is this sequence is homologous to this sequence on the Y chromosome. So those are regions where they act like homologous chromosomes. They have genes in common there. So those pseudo-autosomal regions, or PARs as they call them for short, are what the X and Y use in order for them to act like homologous chromosomes. And here's where the SRY gene is located, up there. A lot of what we know about the X and Y actually came from studying people who have abnormalities in sexual development. So, for example, in your book they talk about, I think it's in your book, they talk about there are some people that have XX, but they're actually male. Or other people who are chromosomally XY and they're actually female. And after doing many studies with them, it was determined that these XX individuals turned into males because they had a piece of the Y chromosome. They had the SRY gene that was incorrectly placed on their X. And so just that SRY gene was enough to turn them into males, even though they had two X's. And then similarly, on these XY females, it turned out that the Y chromosome they were carrying was missing the SRY gene. And so because of that, they turned into females, even though they are XY. So a lot of that information I just told you from originated from studies like that. But you can get a whole lot of weirdness coming out of this. So just really quick, that SRY gene, like I said, is responsible for turning on, or for creating a transcription factor, which would turn on male-specific genes. And so it turns on these genes in cells that are not yet mature, that are in structures that are not yet fully developed, right? Like in your gonads. So when they get turned on, those gonads are going to start to turn into testes, and some of the cells within the testes are, for example, these cells are going to start to turn on or synthesize anti-Müllerian hormone, which will destroy the Müllerian ducts, which you need to do. And then some other cells that get stimulated by this SRY gene, they start to produce testosterone. And testosterone is important for, it's a signal, it's a hormone that has receptors that will turn on development of internal male structures. So the epididymis, which is the structure that comes right out of the testes, the vas deferens, the seminal vesicles, which secrete substances for the semen, etc. Testosterone also gets converted into dihydrotestosterone by an enzyme, And that hormone, right, that hormone also has its own receptors in other cells, and that hormone is responsible for stimulating the development of the male external structures. So the penis, the scrotum, the prostate, and the urethra. So these two hormones are very, very important, testosterone and dihydrotestosterone. And of course, they're stimulated by the SRY gene. Ultimately, that's what they were stimulated by. But you can get a whole variety of other mutations happening that could lead to an individual with ambiguous sex. So for example, if somebody is missing the 5-alpha reductase gene, right? So the one that is responsible for making this enzyme, those people will stop here. They will develop internal male structures, but they won't develop external male structures. So outside they'll look like a female, inside they'll look like a male. Another syndrome or problem that we see happening in some individuals is when they are insensitive to androgens, to these male hormones. So they do not make the receptors for dihydrotestosterone and testosterone. So they develop, or they look like females. And actually they talk about this particular problem in your book. So you can see how this whole development, which is kicked off by that SRY gene, is very, very important for normal sexual development. As you can imagine, doing studies with humans for studying sex-linked inheritance is pretty difficult. You'd have to make the humans have a lot of kids, etc., to do these studies. So instead, a lot of what we know about sex-linked inheritance patterns comes from studying Drosophila melanogaster, the common fruit fly. That has been the model organism. And Thomas Hunt Morgan was the scientist who did a lot of these studies to uncover these patterns. And just to give you a little quick brush up on nomenclature with fruit flies, so for the Y chromosome, we just write Y, right? You write the Y when you want to keep track of the Y chromosome. When you keep track of the X chromosome, you write X, and whatever gene you're talking about, you're going to make it a superscript. So for example, if we're going to talk about the W gene, if it's a mutant allele, we'll make it lowercase and we'll put it subscript, right? If it's the wild type allele, we'll still make it what you would write for the mutant, but we'll put a plus. Oops. So that would designate that that's the wild type allele on the X chromosome. And then when you're talking about the full genotype, right, because they have to represent both of their sex chromosomes. Here, this would be so the mutant allele, little w, means you're going to have white eyes. The wild type is red eyes. And you can see that in these pictures on here on the left. So this individual is a white-eyed male. This individual is a red-eyed female. And you can tell that because she's heterozygous, but she's got a wild-type dominant copy, and you know it's dominant because the mutant's recessive. And then this individual right here is the same thing, a red-eyed female. Okay, so that's just a little bit about the nomenclature. Oops. And so the interesting thing about sex-linked inheritance is the following. So if I took a white-eyed male and I crossed it with a homozygous wild type or a homozygous red-eyed female, what would I get? Well, you can do the Punnett square. So let's put the male over here. Let's put the female over here. Okay, so if this egg met this sperm, we would get this individual. That fly would be a red-eyed female. This would also be a red-eyed female. Okay, so what we would get back would be all red-eyed offspring, really. You'd get, let's see, 50% of them, oops, will be red-eyed females, and 50% of them will be red-eyed males. Right? Here's my males. They're all red eyes, and the tops are the females. Okay, but this is our parental generation. This is my F1. What would we get with our F2 F2? So if we took one of the females, and you can see that both of my females are this genotype. They're both heterozygous red eyes, and we took one of the males, which are all carrying the wild type allele, and we crossed them together. So this will give me my F2 generation. What should I get? So put the female possible gametes there, the male possibilities here, and then when we combined, this one will be a red-eyed female, this will be a red-eyed male. Oops. And then here we would get a red-eyed female, and here we would get a white-eyed male. This is new. So now what we're going to get back for our phenotypes, we're going to get back, let's see, of my females, we're going to have, let's see, 50% will be red-eyed females, 25% will be red-eyed males, and 25% will be white-eyed males. Okay, so what I want you to start noticing is that for sex-linked inheritance, you start to see a difference in what you get for the females versus the males, and we definitely see a difference here. Not only that, but if you look here at the original cross, here we did a white-eyed male crossed with the red-eyed female. If we did the reciprocal cross, and a reciprocal cross would mean we would take a red-eyed male and cross it with a white-eyed female. Let's do that. You're going to see you're going to get different results. Okay, so I said for the reciprocal cross, we're going to take a red-eyed female, white-eyed female, because last time we had a white-eyed male, and we're going to cross her, so she has to have two recessive alleles, and we're going to cross her with a red-eyed male. So this is what they call a reciprocal cross, because it's the flip of what we just did. Instead of a white-eyed male, we have a white-eyed female, and instead of a red-eyed female, we have a red-eyed male. Okay, so let's see what we would get. So I've set it up. I put the female on one side of the Punnett square and the male on the other side, and let's look at what the possible offspring are for this F1 generation. So that's going to be a red-eyed female. This will also be a red-eyed female. Over here, we're getting white-eyed males. See that? Already something different from the previous cross. So our phenotypic results here for the F1, this is our parental, are going to be, we're going to get 50% red-eyed females for all of our offspring here. 50% are going to be red-eyed females, and 50% are going to be white-eyed males. So last time for the F1, we had 50% red-eyed females and 50% red-eyed males. So this is already different, like I said. And so if we take one female from here and one male from this result to do the F2, that means we're going to have this cross. Okay, a red-eyed female crossed with a white-eyed male. And let's see what we would get. So I've placed the parents from this cross here onto our Punnett square. And so this would be a red-eyed female. this would be a white-eyed female. That's new, right? And then we'll get a red-eyed male and a white-eyed male. So now we get, again, for the F2, something totally different than what we got with the other cross, with the reciprocal cross. Here we're going to get 25% are going to be red-eyed females, and 25% are going to be white-eyed females. And then 25% are going to be red-eyed males. And 25% are going to be white-eyed males. This is going to be white. Totally different from the reciprocal cross. So this is just done to show you, I wanted this to show you that if you were to do crosses, like not knowing the genotype, you're just taking a red-eyed male or white-eyed female and you're crossing them, and you keep seeing these differences, these different frequencies and red-eyed or white-eyed between males and females, that should be a huge clue to you that there is some sort of sex-linked inheritance pattern going on. So here is the general rule. For sex-linked single-gene inheritance, phenotypic ratios will look different between the sexes, which you just saw. And number 2, phenotypic ratios look different in reciprocal crosses. So again, what I mean by that is in one cross you did red-eyed male, white-eyed female. In the other cross you flipped the phenotypes between the sexes, white-eyed male, red-eyed female. So the ratios ended up different depending on which cross you were doing.